Neurons are electrically excitable cells that communicate with each other using two types of electrical signals: graded potentials that are used for short distance communication and action potentials that allow for communication over long distances in the body. When an action potential occurs in a neuron or a nerve cell, it's called a nerve action potential, or commonly just a nerve impulse. So to understand the function of graded potentials and action potentials, let's think about how the nervous system allows you to feel that pen that you're taking notes with. So as you touch the pen, a graded potential develops in a sensory receptor over the skin of the fingers. The second step, the graded potential, triggers the axon of the sensory neuron to form an action impulse or a nerve impulse that travels along the axon into the central nervous system and then causes the release of a neurotransmitter. Now, we hadn't talked a lot about neurotransmitters yet, but remember, a neurotransmitter is just a chemical that's released by a neuron that's going to trigger another cell to do something. So the neurotransmitter is released at the synapse with an interneuron. Think back to what we just talked about. The inner neuron was in the central nervous system and it was the processor. So the neurotransmitter is going to stimulate the inner neuron to form a graded potential in its dendrites and cell bodies. Now, a graded potential is just a short little communication over a short distance, okay? So now, in response to that graded potential, the axon of the interneuron forms an action potential. The nerve action potential travels along the axon and results in another neurotransmitter being released at the synapse with another interneuron. All right, so let's think about what we've got so far. The sensory neuron had an action potential that stimulated an interneuron in the central nervous system that stimulated another interneuron in the central nervous system. Okay, with me so far? All right, so this process of neurotransmitter released at the synapse, or the space where the cells join to each other, is followed by the formation of a graded potential, and then a nerve action potential occurs over and over as interneurons in the higher part of the brain have to process, right? So once the interneurons in the cerebral cortex, or the outer part of the brain, are activated, that's when we start having perception. Okay, so let's think back through this again. We had a stimulus out here, and that was the touching of the pen. So that traveled along the axon of the sensory neuron, and then in the brain, in the central nervous system, interneurons started to process that information. So, oh, it's round, and it's long, and it's cylindrical, and it has a cap on it, and it has an end on it. Oh, I know what that is. That's an ink pen I'm going to use to take notes. So all of that was happening in the higher areas of the brain in the cerebral cortex. So all of that perception was processed or integrated, associated through those interneurons. Okay? Now, suppose that you want to use that pen to take notes. So here's how the nervous system would continue this pathway. Okay, so now we're at number 6, right? So we processed all this up here in the brain. So those short little graded potentials took that information into the brain where we're processing all of that with the interneurons in the central nervous system still. Okay, so the stimulus in the brain caused a graded potential to form in the dendrites and the cell bodies of a motor neuron, which is a type of neuron that is going to cause an action to happen in skeletal muscle. So that graded potential subsequently caused a nerve action potential to occur in that motor neuron, followed by neurotransmitter release. The neurotransmitter then generated a graded potential in another motor neuron that then stimulates those muscles to move. Okay? So the muscle cell has to depolarize and then create the contraction. So you talked about this, of course, back in the previous unit. So all of this is happening every time you pick up a pen, every time you feel that pen in your fingers, every time you process subconsciously in your brain what a pen is, and then create the action that allows you to use that pen. So remember back to the three functions we talked about earlier. We've got sensory, then integrative and processing, and then motor, which was action. Okay, so this required that we have small little graded potentials that communicate over short distances, and then longer communication pathways that cause action potentials to occur. So all of this happens whether it is an action potential or a graded potential based on two features of the cell membrane of these excitable cells, right? The neurons. So those two features are the existence of a resting membrane potential and the presence of ion channels. Okay, so right here at this point, I want to suggest that if you don't remember resting membrane potential and ion channels that we talked about back during muscle contraction, this would be a good time to go back and review those, because understanding the action potentials in a nerve cell is going to be dependent on you understanding those terms, resting membrane potential and action potential and ions and these channels. Now we'll look briefly at these again, but if you feel like this is a time to review, I would very strongly suggest that you do so. Okay, if you're still with me or back with me, I assume now that you have reviewed or you're comfortable with the ideas of ion channels and depolarization and action potentials. So graded potentials and action potentials occur because the membranes of neurons contain many different kinds of ion channels that can open or close in response to a specific stimulus. So when ion channels are open, they allow specific ions to move across the cell membranes down their electrochemical gradient. That's simply a concentration chemical difference plus an electrical difference. Now remember that ions move from higher concentration to lower concentration, so that's the chemical part of the gradient, but also positively charged cations move toward negatively charged areas, and negatively charged anions move toward positively charged areas. So that's the electrical aspect of the gradient. As these ions move, they create a flow of electrical current that can change the membrane potential. So ion channels open and close due to the presence of gates. So we'll look at some of these types of gates, such as the passive leakage channels that are not necessarily gates, but they simply allow ions to move in and out of a cell. So they're randomly open and closed, but mostly open. And they can be selective in what they allow to pass across the membranes. Active or gated channels have to be opened because they're ion specific, and something has to change in order for them to open. So chemically gated active channels excuse me, are going to be stimulated to open by neurotransmitters. Voltage gated will be stimulated to open when that membrane potential changes, and mechanically gated channels will open because of some type of pressure or vibration, or something stimulates mechanically those receptors. So on the next slide here we have just an overview of these types of channels. So let's go briefly through those. So the leakage channels randomly open and close, but are mostly open, found in most all cells. Your ligand gated, ligand is a chemical such as acetylcholine. When it binds to those receptors, then the receptors are usually going to be a part of those gates or nearby those gates, and it will stimulate the proteins to change shape and the channels open. Mechanically gated are gates that respond to a change in the shape of something. So They respond to a mechanical stimulus such as pressure or touch or vibration, or when tissues are stretching and your voltage gated will open in response to a change in that membrane potential. So all of these types of channels are going to be responsible for maintaining or changing the electrical activity of that cell by allowing or not allowing ions to flow into the cell. Like most of the cells in the body, the membrane of excitable cells, and of course we're talking about neurons, is going to have a membrane potential, which is an electrical potential difference, voltage difference across the membrane. In excitable cells, this voltage is termed the resting membrane potential. Sometimes you'll see me abbreviate that as RMP. So this membrane potential is sort of like the voltage stored in a battery. So if you connect the positive and the negative terminals of a battery with a piece of wire, electrons flow along the wire. So this flow of charged particles is called a current. In living cells in the human body, this flow of ions rather than electrons is what constitutes the electrical current. So the resting membrane potential is established in cells because of a difference in charge on either side of that membrane. So we have a small buildup of negative ions inside the cell along the membrane, and an equal buildup of positive ions along the extracellular fluid on the outside of that membrane. So this difference in positive and electrical charges gives us a potential energy, which we measure in millivolts. So if we look at what's going on inside and outside the cell in this image, you can see that overall we have a pretty even charge inside and outside the cell, but we do have right along the edges of the cell a lot more positively charged ions in the extracellular fluid and a lot more negatively charged ions right along the membrane in the intracellular fluid. So this unequal distribution of the ions results in that ability to change the charges and then use the potential within that cell. So the creation of the resting membrane potential is there because of that difference in charges. I often say resting membrane potential. When the cell is at rest, it has the potential to do its job. So to fire an action impulse because of that difference in charges. So a couple of additional things to bring up here. Most of the anions, or the negatively charged ions, don't leave the cell, so that also helps to maintain that electronegativity on the inside of the cell. And the cations, the positively charged ion concentration, is maintained with sodium and potassium pumps primarily. So remember, cells that have this difference in charge across the cell membrane are said to be polarized, right? So one side of the membrane is more positively charged, one side of the membrane is more negatively charged.